Okay, the good news is I think I figured out why my pictures weren't very clear, so hopefully that'll be fixed. So the question is, how can we activate Krebs? And the answer is a couple of different mechanisms, right? Krebs activation, or really Krebs phosphorylation, right, can occur via pKa. And remember the signaling pathway for pKa. In order to activate pKa, cyclic AMP was generated from ATP, right? So ATP is converted to cyclic AMP by the enzyme adenyl cyclase. And it was a heterotrimeric G protein, right, as we work our way backwards, the G protein that activates the enzyme. And so that required a ligand all the way upstream to bind to a G protein coupled receptor. That's one of the things that can activate Krebs. Another thing that can activate Krebs is here we see a receptor tyrosine kinase, an RTK. A second thing that can activate Krebs, this works, right, by activating, this is RAS, I don't know if you can see this. But so then RAS goes on to activate, and they only show MAP kinase here, right? This is just MAP kinase. And they don't show MAP kinase, which activates MAP kinase, which activates MAP kinase. In either case, right, pKa or MAP kinase are both able to phosphorylate Krebs by entering the nucleus. Okay, so that's how they bring forward their message. Another way in which the Krebs can be induced or activated is via increases in intercellular calcium. Okay, and so the Krebs right here is bound to a site on DNA called Cre. So Cre is the DNA, right? So it's a section of DNA, the DNA binding site. Oh God, that was bad. And if Krebs was cyclic AMP response element binding protein, Cre is cyclic AMP response element, right? The element being the DNA. Now, you can see that it's Cre/Care in the picture. And so when Krebs is activated in a calcium-dependent manner, right? You can see that instead of Cre, it's associated with the same site, only that site is now called Care. Okay, Care is calcium response element. So the Cre is the DNA binding site / Care. Same idea, right? So Krebs is bound to DNA. Where does it bind DNA? On either Cre, right, depending on if this requires calcium, or Care, depending on if it doesn't, right? So what the DNA is sort of the name that it's associated with tells you if it's a calcium-dependent process. If Cre is associated with Krebs, it doesn't require calcium. If the Krebs is associated with Care, if you see that in, say, a schematic or a diagram or an exam, you know that calcium is going to be required, right? So Care is calcium response element. It's a lot of song and dance for this idea where opening up a calcium channel, right? So here's your calcium channel, which allows calcium to flow in. These calcium ions can make their way into the nucleus and activate a kinase. Now, I think what should be here that's not shown in this picture is calmodulin. Right, so the calcium will form a complex with calmodulin and activate a CAM kinase. So CAM is really what enters here and activates something called CAM kinase 4. It's basically CAM kinase 2, or part of that same family. Okay, and so an increase in IC, intracellular calcium, right, leads to activation of CAMK4. Not a big deal, just think of it like CAMK2, and that results in the phosphorylation of Krebs. So what's the big deal with phosphorylation of Krebs? What does it lead to? Transcription. Transcription of what? Multiple different genes. Genes that may code enzymes, structural proteins, ion channels. Okay, so all of these proteins will be expressed, and their transcription is under the control of the transcription factor Krebs. Right, and so you see RNA polymerase associated here only when the Krebs has been phosphorylated. Otherwise, this transcriptional complex that includes RNA polymerase cannot bind. I don't have a slide for this particular effect because it's pretty short with respect to its description. Regulation of transcription can happen via nuclear receptors. So if Krebs was our first type of nuclear signaling, nuclear receptors would be our second. And so the idea here is it requires, right, nuclear receptors require a membrane permeable ligand. So rather than having to use a receptor that's on the surface of the cell, the receptor is actually nuclear. Okay. These nuclear receptors, when they're bound to the ligand, act as transcription factors. So the nuclear receptor binds the ligand. Right, this is a transcription factor. Now it works as a transcription factor. Typically the receptor is in the cytoplasm and translocates into the nucleus upon ligand binding. So the receptor in cytoplasm the receptor ligand complex, right, so when they're coupled together, right, it moves into the nucleus so it can have its transcriptional regulatory activity. So this is now moving, translocates into the nucleus. An example of this would be a hormone like cortisol. Okay, cortisol is a glucocorticoid, so this might be something that you've seen in anatomy. So it's going to regulate levels of glucose. So an example would be cortisol. It's a stress hormone, so we know it's released in periods of stress. In any case, right, when it binds to its receptor, the receptor unfolds. So unfolding of the receptor, right? It's just the receptor's a protein, right? And so it can come apart, right? Conformational change into the nucleus. And affect transcription. All right. The third and last example we'll talk about with respect to nuclear signaling is something called CFOS. CFOS is a transcription factor transcription factor. Okay. And it's present typically at really low levels, right? So as CFOS is used to tell whether neurons are mature. And so in immature neurons, there's very low CFOS. Oh boy, I knew that was going to be bad. Immature is the word I'm going for. Okay. But induction of the CFOS gene causes it to go way up, and its stimulus of the gene happens directly. Okay, so stimulate the cell. Right, that causes transcription of CFOS gene. Which causes the CFOS levels to spike. So CFOS protein goes way up. Okay. And CFOS then induces the synthesis of many other genes, right? Because itself is a transcription factor. So now that there's a lot of CFOS protein present, it induces multiple other genes. Many of which are involved in development of neurons, right? So things like proliferation or transformation of cells from their stem cell-like state, okay? So CFOS, the textbook uses this term, and you'll see it sometimes, right? It's considered an immediate early gene, right? The CFOS gene is. Because it's one of the first genes that gets turned on as neurons are developing, now other genes, right, that are downstream of CFOS, right, these multiple other genes, these genes are called delayed response genes because they require the CFOS gene be activated first. Okay, so that's how CFOS signals and why it can be used as a marker of development. Okay. So let's talk about how it's activated with respect to our cell signaling conversation. So CFOS induction, that is the turning on of CFOS gene. All right, so to get CFOS protein present, the one that works as a transcription factor, requires convergence of multiple different pathways. And let's talk about what some of those pathways are, but I'm going to do that in the next video. Okay, and I might try to find a better picture that shows more conversion. I pulled this from a different textbook, but I think I have a better picture for you. So stay tuned.